In this video, we are discussing the topic of the Western blot. A Western blot can be used to specifically detect proteins. The term Western blot is a play on the previously developed Savan blot, named after Edward Savan. The term blotting comes from the blotting paper used in the original method to transfer the analytes. Several techniques have evolved from the original Savan blot method. In the Western blot, proteins are detected using antibodies, which are also proteins. In this course, we will also learn about the Northern blot technique. To learn more about Northern blot, refer to the corresponding video. The execution of a Western blot can be divided into 4 main steps: the protein separation, transfer or blotting, detection, and visualization. The following slides will provide more detailed explanations for each of these steps. For Western blotting, it is first necessary to separate the protein mixture under investigation. SDS page is suitable for this purpose. During SDS page, proteins are separated by size in a polyacrylamide gel through electrophoresis. For more detail, refer to the SDS page video. After separating the protein mixture, peptides are transferred or blotted onto a membrane. There are various methods for this, including blotting in a blotting chamber, a type of electrophoresis chamber, or using a semi-dry blotting technique in specially designed devices. Since this course uses the semi-dry blotting technique, this method will be discussed. A stack or a sandwich is created consisting of filter paper, membrane, gel, and more filter paper. All components are soaked in a transfer buffer and layered in the chamber. When an electrical voltage is applied perpendicular to the gel's running direction, the protein bands are transferred from the gel onto the membrane. The pattern of the separated protein bands remains intact. The blotted proteins can then be treated for immunodetection in the next step. After successfully blotting the protein bands onto the membrane, proteins can be specifically detected using antibodies. Typically, both a primary and a secondary antibody are used. The primary antibody binds to the target protein's antigen, which may be, for example, a 6-fold His tag. The secondary antibody recognizes the primary antibody as an antigen and binds to it. The secondary antibody is modified to enable visualization of the target proteins. It is often linked to an enzyme or labeled with fluorescence. In the past, antibodies were also labeled with radioactive markers. Merely incubating the membrane with antibodies is insufficient, because non-specifically binding antibodies can lead to a false positive signal. Therefore, blocking non-specific binding sites is crucial. This step, known as blocking, involves incubating the membrane with proteins, for example milk proteins in a buffer solution, to block non-specific binding sites on the membrane. Afterwards, the primary antibody is applied to bind the target protein. It is essential to perform a washing step before applying the secondary antibody to remove excess primary antibodies. This washing is also done after incubating with the secondary antibody. In this course, the secondary antibody is covalently linked to the enzyme alkaline phosphatase. Proteins can be identified on the Western blot by adding chromogenic or luminogenic substrates for the covalently bound enzyme, resulting in a color precipitate or luminescence. In this case, alkaline phosphatase catalyzes the formation of a purple precipitate. A BCIP and BT solution is used as the substrate. When BCIP is dephosphorylated, the reaction product reacts with the soluble yellow NBT, forming 2 insoluble substances in the form of a precipitate on the membrane.